large, like it was almost like a football. Big expanse. I had all these palm trees, all snow.
Thank you so much for coming. Yeah,
I think that's hilarious.
I think, well, of course, I
And
grew up by
our
it, right?
other neighbors,
We're actually
their
getting
houses,
started on
big
time,
snow
which
pieces,
20
same
minutes
thing.
ago, it did not look like this was going to happen. So today, I want to welcome everybody to the Scholar Series, the first one of this semester. We have another one next month, going to be Professor Rob Kipka, and the date and title and time and all that stuff will be sent out soon. We'll announce it that way. We have a couple more talks coming up, but look for those to be announced, and it'll all be good. But we're really excited about this. So I have to tell you that I've been wanting to get Professor Ranson Olsen up here as part of our speaker series for a long time now. I've been here 10 years, and I have to tell you, she's always been this wonderful mix of really super smart, but really approachable and friendly. So like she's got this wonderful combination of what I think makes a fantastic, faculty member and colleague. So I'm happy that we've been able to finally book her. And normally I give a long, intro about where you went to school and all those wonderful things. But you're going to do that for me, right? You're just going to make it easy on me.
Yeah.
So everybody, help me welcome Professor Britt Ranson Olsen.
I muted. No, here. Oh, well, thank you so much, Mark. And thank you, the library staff, for recruiting me to do this. I know you asked me last year, and that was so busy, right, to find the time. But yeah, no, I wanted to do this. I'm very excited to be here. Thank you all, for coming and attending. It being a scholarly series, I felt a little bit of pressure, right, to talk on a subject that I knew, pretty well. And so, regarding like my background and such, I was a biology major here at Lake State, so fellow graduate, many of you soon will be. And then I got my master's degree from Michigan Tech University, also in biology. And then I received my doctorate in biochem and molecular biology at Oakland University. So I came back. I was lucky enough to come right back after graduation and start teaching human anatomy and physiology. So I've been teaching that for 17 years now. And one of my other classes and love is cell and molecular biology. So this is the topic, right, that I selected basically. Took a lot of time to get updated on, and feel that it's a pretty interesting topic. So I hope you enjoy it. It's all about twins, right? And specifically using twins as an experimental model to study epigenetics. So I started off with this particular picture. This is the first developmental stage of twins forming. So they occur when the embryo spontaneously splits into two. This is identical twins. And as a result, we're going to see you produce genetic clones of one another. So they share 100% of the same DNA. Okay, so I thought I would start off with one of the first slides that I use in my cell biology class. I generally start off the students, they're upper level, usually science majors, with a reminder of what the basic properties of a cell are. And one of those we see is the ability for cells to respond to signals and to be aware of their environment, respond accordingly. So this was a classic, very old experiment you see over 100 years ago, in which they used sea urchin to test and observe development. And what they did was they separated the very early embryonic stages, artificially separated those cells. And what they found was that if it was early enough, if it was only at the 2 or the 4 cell stage, then each of those individual cells could further develop and form a complete whole organism. And so, obviously, why that's cool and what it tells you is that each of those very early cells has all of the information necessary for developing and becoming any of, the kind of adult, fully developed tissue types. And they, basically created little artificial, twins in this case. Okay, so these are the developmental stages, again, what occur once the egg gets fertilized. And what you can see is that the first stage in fertilization is the zygote. So that's where you're actually combining the maternal and paternal DNA. And once that happens, then the cell begins to divide. So from 1 to 2 and 2 to 4. And like what we saw with the sea urchin experiment, when it's at those early stages, again, it has the structure has the full potential, right, to develop into a complete organism. So it's what we call totipotent. Totipotent, if you kind of like break down the word, I do that in anatomy all the time, right? Potent, think potential. Toti, think total. Okay, so the total potential to develop into an entire organism. Beyond that, into the 8 stage cell, the cells continue to multiply when they form a solid ball of cells. This is what we call a morula. And then from there, we develop a hollow ball of cells, which is called the blastocyst. These latter stages are what we classify as pluripotent. So they have the potential to develop into multiple or plural adult tissue types, but not the whole body, like not the whole thing. So they're a little bit more, locked into what their fate is going to be. And that's pretty much how it goes. The older a cell gets, generally the more and more locked into its identity. What was I going to say? With the pregnancy, where the implantation happens in the uterine wall, it's usually in one of these latter stages. Okay, so like we're talking, right, obviously about twins. So we want to look at how identical twins form. They're termed monozygotic because they develop from one egg. So one sperm fertilizes one egg, and then what you see is that there's a range, right, a period of time in which that embryonic stage will spontaneously split. So early, up to, we see 3 days post-fertilization, all the way up to 12 days post-fertilization. If those structures split, the results will be these, identical twins or a twin pair. Depending upon when the split occurs, what you see are different maternal or structures of pregnancy. So if it's very early on, each of the twins generally has its own placenta and its own amnion. The later the split occurs, you see that they oftentimes will share the structures. So in this case, they are monoamnionic, monochorionic, one placenta, one amnion. Beyond the 12 days, if the split were to occur late, like day 13 or more, then there's the chance for a lack of complete separation. So that's where the conjoined twin factor kind of comes in. So these are monozygotic, these are identical twins. If we compare those to how fraternal twins develop, you can see first off they are dizygotic. So in other words, it's 2 separate eggs that get fertilized by 2 separate sperm. And those 2 structures, they're separate, they remain separate through the remainder of the pregnancy. They just happen to develop along the same time and generally, are delivered close to the same time. And in the case of fraternal twins, you see that they always have their independent structures. The whole process is independent. So they each have a placenta, they each have the amnion. Okay, so twin studies have been carried out for decades. They are so useful as a means to decipher between genetic influences of certain like physical traits, but also
also disease states and such, the genetic influence over the environmental influence. Okay, so in the case of twins, you just imagine most of those, especially early on in life, are going to have the same environmental, right, factors affecting them, the same exposures, maybe, the same food availability and such. So to compare, let's see, might be a little hard to see. The identical twin are in white, the fraternal twins are in the gray. What you can see is this comparison of traits that those twin pairs have in common. Those up at the top are more of the phenotypic traits, such as height, but also you see reading, disability, autism. All of those tend to have a greater genetic influence. And we see that based upon the identical twins, the monozygotic twins, having a greater percentage of that shared trait. Those factors down at the bottom, these are like more complex diseases, cancer and stroke and such. Those tend to overall have more of an environmental influence, which I think makes sense because those are the conditions that most often occur or erupt later in life, so they're more of a result of a buildup of exposures. Another thing that you want to pay attention to when comparing these rates is that in those traits where there are larger discrepancies between the percent seen in the identical twin versus the fraternal twins, that really tells you something, okay? So just to kind of reiterate that or look at similar data but just from a different viewpoint, both the dizygotic and the monozygotic, so fraternal and identical twins, they're both likely from a similar environment. Again, if we're looking at the early years. If you see a larger percent shared trait in the identical twins compared to the dizygotic twins, again, living in the same environment, then odds are there is a much stronger genetic factor. And so a great example of that is autism. If you look at the percent shared rate of the identical twin here versus the dizygotic twin there, you see that there's a drastic change, right? That tells you that there's a strong genetic component there. And again, the more complex diseases down at the bottom, not so much, again, more of an environmental effect. Okay, so this is a look over the last several decades at the birth rate of twins. This is per 1,000 births in the United States. And what you see is a pretty steady increase. It kind of peaks out, plateaus out here in the 2010s, and it's more recently kind of settled down a little bit. And the same thing I found in Australia, where they've been monitoring it as well. I was going to ask you, the attendees, why do you think this is what might contribute in the 2000s and the 2010s to there being more twins being born? Yeah.
Does it have something to do with like in vitro fertilization,
Yes.
stuff like that?
So IVF, absolutely, you got it. Anything else? Yes, good, maternal age. Those are the two biggies. Yes, absolutely, absolutely.
Here's to older mothers.
Yes, we're very mature, right?
Yeah, and really old fathers.
No, those were my exact two reasons, IVF, right, and increase in maternal age during those periods of time. The interesting thing, though, is that both of those things have nothing to do with identical twins. Okay, the formation of identical twins is a spontaneous event. It's only the fraternal twins that have a connection with IVF much more strongly and maternal age. And what it boils down to is the older the mom, the more tendency to ovulate more than one egg per cycle. And IVF, there's a clearer connection between increased ovulation rates, but not so much with, again, the actual identical twins.
So this is both kinds of twins?
This is both kinds That's right. Yep. Good. Do twins run in families? Yes. Mark,
Yes,
I
they
love.
must, because the two people I know in here with twins, me and my wife, both have twins running in our family.
In your family. Okay. Are they, do you know, are they fraternal or identical?
Well, I know that Mark has nothing to do with our twins because I know how biology works. But
Okay.
I have nothing to do with it.
Oh, goodness. I love.
no, but my mom had twins when she was 36, and I had twins
Okay.
when I was 36.
When you're 30.
And that could be a complete coincidence. I hate it when people say it skips a generation because I don't think that that's a thing.
Yeah, yeah. So,
I think it's an age thing.
so the only time this applies, do twins run in your family, is if it's in the case of fraternal twins. So the non-identical twins, there is a gene, right, that moms can carry that causes them essentially to hyperovulate, be more sensitive to hormones and hyperovulate throughout their reproductive years. But again, that's only in effect on the fraternal, so the dizygotic. This does not apply to identical twin formation at all. Again, completely spontaneous, right? Like an artifact of nature. Okay, so I told you I had to talk about something that I felt I knew well, right? To do a scholarly series, a scholarly talk. One of the reasons I chose twins is because I'm also the mother of twins, right? I'm shocked and awed to find two embryos, oh my God, on the ultrasound. Yes, so this is Henry and Thomas, so Walker, Texas Ranger. This was like our Talladega Nights phase. I did not allow them to drink moon mist. It was for just for the picture, right, is all. But yeah, we were shocked. I hadn't done IVF. We have a set of twins that we're aware of in our family, but we have huge families. So it really didn't run in our family. They were both males. We found that out. The structures did not determine one way or the other because they each had a placenta, they each had an amniotic sac, so it could have gone either way. So we waited till they were born. They both had the same blood type, A positive. So it's like, oh, what are we doing? I had to know, like, right? With obviously just being their mom, having that innate interest, but being a professor in this area was something that, well, yours are really cute too. There's something, that I just had to know. Shortly after they were born, we swabbed their cheeks and sent away their DNA and had the zygosity testing done. And what that basically is like a DNA fingerprinting analysis. Where you can basically identify a human, distinguish one from the other with a very high statistical output. And these were their results. I should tell you, I got the results back. And they said that there was a 99.998% chance that they were identical. They were monozygotic, so we had identical twins. And then I reached back to the company and I explained to them that I was a biology professor, and they were very kind. They sent me the raw data so I could use it in class, which I do all the time. And what you can see, I don't know who's who. This one boy, this is the other boy. They didn't tell me that. But the black, the blue, and the green peaks are all unique identifiers. These are all patterns, just like a fingerprint. And what you can see is where the peaks come off on this x-axis it's forming an identical pattern. Okay, so that again, without a doubt, we determined that they were identical. So yeah, very cool. Henry left, I think Thomas, right? Okay, so Thomas and Henry. When they were born, there were amazing similarities between them. There still are. These things come about, and it has just been it's just been impressive to be able to observe them growing up and to watch this and I guess appreciate it from a cell biology level. But there have also been a lot of differences that have, developed and kind of, occurred as they've grown up. They have different preferences. Henry for sports, Thomas for art. He'll sit down and draw a whole picture. Henry would never have the patience to do that. Preferences for food. Thomas loves Brussels sprouts. Henry hates them. You start kind of thinking, well, how can that possibly be? These kids have the exact same environment, right? Same household, same dinner, everything, same exposures. How is it that they're exhibiting these different traits? And then, like I said, as they get older, you see even more differences. And one of the ones that really hit home with me was when Henry was clearly right-handed and Thomas was left-handed. That's like your brain's wiring, right? And they're genetic clones.
And their hair as well.
Their hair, do you have like the cowlick thing going the opposite way? Oh, mine, that's right. Wow, so you've probably noticed something that I haven't. Okay, right, yes.
Do I tell them apart?
Well, so that's how you tell them apart.
No, one of them has a freckle on his hand.
Oh, yeah,
And
okay. So she teaches music. This is their music teacher at Lincoln, so I'll have to look for that. All right. So
they're mirror image.
they might be mirror image. Yeah,
I
yeah.
just pretend they are.
Yeah, no, that it could potentially be. There's evidence that supports that, but then there's evidence that, yeah, I don't know. So if you have your own twins, right, or if you've ever known twins or have twin relatives that are identical, you realize that there are subtle differences. So identical twins, despite having the same DNA, they're genetic clones, they are not 100% the same. So really that raises the question, how can that possibly be? Especially when they're also being raised in the same environment. Okay, so one of the likely answers to that is what is called epigenetics. Okay, so genetics, that's of course all of our DNA and all of the instructions and information that our cells rely on to establish ourselves and keep us healthy. Epigenetics, the epi, remember in anatomy, means above or on top of, and the genetics meaning on top of the genes. Epigenetics are really subtle chemical modifications that are made on top of the DNA. And what the outcome is that they affect how we regulate those gene sequences. So in other words, whether we express a gene or not, epigenetics can completely silence a gene. It can shut it off, but it can also turn it on and produce a lot of whatever that gene message is. So what basically initiates epigenetics is your environment. So it's everything that we're exposed to, everything that we inhale, absorb, ingest. Diet is a big component of that. What pharmaceuticals we're on, our habits, right? Our good habits and our bad habits. All of those things are known contributors to epigenetics. Okay, so this is, I think, a good schematic. It shows a cell and all of its DNA kind of spilled out for us. So the DNA, double helix, hopefully that's familiar to you. You can see in areas along the length of the DNA, it's wrapped around, these are organizing proteins called histones. In this particular image, they've tagged the epigenetic marks with these green and yellow fluorescent tags. So you can see again that the chemical modifications are actually on the DNA. The gene sequence is not affected at all. Your genetic message remains the same. Okay, so to explain how all of this works, like you saw on the last slide, there's a lot of DNA in each of our cells. There's approximately 6 feet that has to fit into, the confines of our nucleus. So that's a very small space. So the DNA, as you find it, is, again, highly organized. You can see up here where it's wrapped around, again, its little organizing protein, the histone. How epigenetics essentially works is by either increasing or decreasing the accessibility of the gene sequence to large protein complexes that are required to express the gene. Okay, so this is an example of an epigenetic effect or modification, and that is the acetylation of those histones. Okay, so in the case of an acetylation, you can see the little acyl group added to the histone there. What that does is it loosens the DNA that is wound around the protein, making it more accessible than to the proteins that have to dock to that sequence and actually migrate along the gene in order to read it. So with acetylation, then that turns that particular gene on. It can be expressed. When you remove the acetyl groups, deacetylate them, you see that the DNA is wound more tightly around the histones. The proteins can't get access to the gene message then, and that essentially shuts the gene off, or what we call silences it, turns it off. So the acetylation is an epigenetic modification. Another modification is methylation. These are like super simplistic chemicals, you guys, like so tiny, and just like carbon, a couple oxygen, some hydrogen. They're like the chemistry is like as simple as it gets. But in this case, we already said if the histone is acetylated, that opens it up for expression. Without methylation, that also promotes gene expression. In this case, you remove the acetyl groups, you lay down a bunch of methyl groups in the gene sequence, and that shuts it down. It's almost like, I guess putting down glue or something. You're not going to get access to, again, that genetic message. Okay, so this was a very early but groundbreaking, I would say, epigenetic study, and it's really cool. Sometimes the more straightforward ones are the best, right? Because they're powerful. What they did was they took what is called an agouti expressing mom. So they used obviously little lab mice. Those mice that express this agouti gene happen to be overweight, so they tend to be obese. They tend to be yellow, so that's the pigmentation. And then they carry all of these downstream traits, such as a higher incidence of diabetes as well as cancer. With reproduction from that mom, you would expect her to, if it was a dominant case, have offspring that also expressed the agouti gene. So they were also yellow, heavy, disease-prone. What they did was they took the same agouti mother and they fed them methylating diets. So these were food groups that contain a lot of methyl groups, and basically those methyl groups were laid down on the agouti gene, and the agouti expression was silenced in those offspring. And as a result, they produced a mouse of brown pigmentation, normal weight, and again, those downstream incidences of cancer and such became, more baseline, more normal. So this is just a great demonstration of how something as little as your diet, right, can have such dramatic effects. But they know what the gene target was in this case. Another early and kind of mouse model study in epigenetics was looking at the expression of the glucocorticoid receptor. So that's what stands for GR here. That is a receptor that ultimately helps govern how we handle stress and anxiety. That's what they measured in this particular model. What's interesting about the glucocorticoid receptor is it's usually silenced already at birth. So all those little red marks are methyl groups. so it's been shut off. Mother has her babies. What they found was if the mother was a nurturer, so if she cared for the babies, if she bathed the babies, took good care of them, what that behavior ultimately resulted in was the removal of the silencing marks. Those babies would express the receptor and have less anxious, more stress-free, right, adaptations. In the case of mothers that neglected their babies, the silencing markers remained. And so these offspring did not produce as much glucocorticoid receptor as over here, and they remained, you see the little face, anxious and stressed. You see, yes. So that's amazing again, where it's looking at or showing us how a behavior, right, a maternal behavior, is again affecting this expression, and therefore really affecting the outcome and potential behaviors, right, of the offspring. Okay, so we've already said, right, that diet has a direct connection to the epigenome. Yeah, you have your folic acid and your omega-3s, but I mean, kind of everything, our environment, our stress levels have been directly linked to changes in epigenetics. Your overall health status, right? Obesity, who we live with, right? Your pets, where we work, you see farming, etc. What we do to our bodies, drinking, smoking, directly linked to changes in epigenetics. And one thing that I feel is very interesting, there's a lot of research potential there, is the link that they've made between our epigenetics, these chemical modifications, and the types of microbes that live in our gut. Okay, so those microbiota, one is affecting the other and vice versa. So they're playing off of one another. So that'll be, yeah, really, really interesting to see what comes of that. Okay, so the topic for this talk was twins and epigenetics. So I had a hard time, you guys, like trying to decide what to include in the talk. Oh my God, there's so much and so interesting. But what I want to direct the remainder to is what twin studies in epigenetics have divulged. And so, actually, this is like a zoomed in picture. One of the things that I noted earlier, and that's been noted in the literature, is that even in identical twins, their differences become greater with time generally. Again, like the phenotypic traits, but also more complex things, they definitely get greater with time. And, to the point where some identical twin pairs in their older years, one might get diagnosed with cancer and the other one doesn't. Or one with chronic hypertension and the other one doesn't, right? These are noted things that happen. How, again, can that be? Well, this study, again, looking at identical twins and their epigenetics might help explain that. What they did was they looked for epigenetic similarities in the chromosomes of 3-year-old pair twins to that of 50-year-old pair twins. So, obviously given a lot of time in between. And what you're looking at, the fluorescing, right, chromosomes, is that in the yellow, these are areas on the chromosomes that have homologous epigenetic marks. So they're the same then. And you can see there's a lot of yellow there. And that's early. That's at 3 years of age. At 50, most of the yellow is eliminated. This is actually green. It's green and red. And those are tagging different epigenetic chemical moieties. I have a more kind of zoomed in view. It's the same information, but just again, one chromosomal pair in the 3-year-old versus 50-year-olds. And again, the yellow shows the homologous epigenetics. And you can make out that pattern, the 4 yellow bands. The 50-year-old twins, there are just little spots of yellow remaining. Okay, so all that they had so similar is almost completely eliminated and now completely unique. So the red and the green are showing different epigenetic tags. So what this tells you is that, number one, epigenetics, they accumulate with age as a whole. And in regards to twin pairs, they diverge those twins, right? They're making them different as the environmental exposures, time, right, and all of that adds up. And it could also be, a contributor again to why one twin later on in life develops some kind of a disease where the other one doesn't. There's a lot of studies, not surprisingly, other diseases, right? In looking at cancer, so epigenetics in oncology. I know that, I'm sorry that this font is very, very tiny, but basically to prevent a healthy body from developing cancer, really two things should be happening. And hopefully they're all happening in us right now, right? We want to activate tumor suppressor genes. So we code for these tumor suppressor genes, which should get activated. They should come to work, to play in the early signs of cancer. That's one thing. The other kind of gene line we want to target are oncogenes. So oncogenes are those genes that we also unfortunately carry, where when they become overactive, they can induce tumor formation. So we want to keep those suppressed or silenced. What happens, unfortunately, but it's good, it's coming to light, with tumor incidence is they're finding that these tumor suppressor genes are being silenced with the epigenomic signatures, whereas oncogenes might be being activated. So that would be the connection, really broad connection between epigenetics and cancer. But with twin studies, what they found in the case of breast cancer, where there's a twin pair where one twin gets diagnosed, the other one doesn't have the cancer, they've been able to identify blood markers. And again, these are epigenetic biomarkers in the blood of the twin that has the cancer. And the beauty of that is that they can look for that similar biomarker, that signature in any body that they, are concerned about. It's very similar to like a PSA, kind of a prostate serum antigen test. And the beauty, again, like you see here, is it's not invasive, and it's such an early means of detection. They can really find out, before the tumor has a chance to really grow. Okay, another most recent identical twin-based study, the NASA study with, these are identical twins, Mark and Scott Kelly. I have a mental block for their names. So let's talk about smarty pants, right? Identical twins, both NASA. What they did was really cool. They sent one brother to the International Space Station. He was there for a year, while the other astronaut remained on the ground. And they measured everything in them and on them and from them. So all the biological samples, all the cognitive, physiology, muscle reflexes, all of that, in order to assess what space does to the body, what is happening there. And they compared the results, right, of the twin that stayed on the ground to the results of the astronaut, well, the one that went up. And amongst, again, all the measures that they took, you see one of those that they looked at were the epigenetics. So again, the chemical signature changes on the astronaut's genome. What they found, and I just honed in on that part of it, was that Scott, so he must have been the one that went up to the ISS, he experienced epigenetic changes in flight, but the rate of the changes were the same that Mark experienced down on the ground. So what it told you, because we talked about those changes accumulating with age, is that the accumulation rate basically remained the same, right? That there maybe wasn't such an effect of being in space. What they also found was that the space exposure did elicit epigenetic changes at slightly different genes, but that wasn't so interesting, really. It wasn't such a big deal. What they found was those that he did accumulate when he was up, they returned to baseline when he got back to Earth. So that was huge, right? That when he came back, his environment changed, and he basically reverted back to, what his brother's was. So that is a very important finding when considering the fact that these epigenetic changes can be reversed. So that's huge for us. Okay, so these are like the two big take-home messages I wanted you to have, because these apply whether you're a twin or not. Most of us aren't, right? Number one, epigenetic modifications can be reversible. And number two, I'm going to leave off showing you that these epigenetic traits, they can actually be inherited. So you can actually pass down your traits to your offspring and beyond, and what the implications of that, really are. So epigenetic changes are reversible. We saw that with the twin NASA study. This is an experiment, again, using the poor little lab mice. These are 10-month-old identical twins, the two little lab specimens. And they obviously, you can see that one is aged, right? Probably far beyond where he should be. What the researchers did was they purposely elicited these epigenetic modifications that they knew would induce aging in that particular rat. And what they found, obviously, you can see it, all the visible stuff, but they saw, that he clearly had deficits in his nervous system, in his muscle activity. He lost a lot of vision. I think they actually referred to him as being blind on some level. But then what they did following, I can't remember the period of time, was that they restored the changes. They purposely removed them or put them back, the epigenetic chemicals, the way where they should have been, right? To mimic, of course, the unmutated mouse. And what they found was that they were able to rejuvenate, again, both the nervous system, the muscle, and much of the animal's vision. So it was very, very cool. In regards to applying this to us and what we can do with this idea that these epigenetic changes are reversible, well, you can start making more healthy changes, right? Replace our bad habits with our good habits. An example of this is smoking. There are clearly defined epigenetic regions in the genome that are heavily methylated in smokers. That's been established for a long time. But what they've demonstrated is that those individuals who are able to quit, this is a year following cessation, is that they're basically able to bring it back to the baseline where the non-smokers were. So I thought that was very incredible, right? Think about what you can do, with your diet, for instance. The last kind of idea, again, I want to leave you with is the idea that epigenetics can be inherited. All our genes can be inherited, obviously, right? But this is a very, I think, critical finding. What we're looking at, male and female, right, organisms, is that if the mother or the father are exposed to some kind of environmental trigger that they know is going to induce an epigenetic modification, they can pass those down to their offspring. That's intergenerational inheritance. But in the case of a mother who's pregnant with a female fetus, not only is the mother and her egg affected, but the eggs that are present within that female fetus can be affected or impacted by that. Females are, different developmentally because in the case of a female, they are going to produce all of the eggs that their body will ever produce through their reproductive years in utero, still in their mommy's tummy. Males are completely different, right? They don't start producing their gametes until they're kind of reproductive age. In the case of a female, passing that epigenetic, they call it epimutation, down not only to the F1 but also to the F2, again, if that initial mom is pregnant. And then from there, some of the epigenetic marks can be lost, but some of them they're finding are still heritable. And then you get into what's called transgenerational inheritance. So really, like the point being here is, what lifestyle, say, your grandmother led, what she ate and drank and smoked when she was pregnant with your parent can potentially impact your genetics and how you express your genes. And the same thing goes for all of you, if you intend on having, a family someday. What you do to your body now can impact those generations to come. And again, they're making these linkages. Okay, so epigenetic modifications are reversible. This is good news. Yay for everyone. All right, lifestyle changes are good, and they can be inherited. Okay, so this is how I begin introducing epigenetics to my cell biology class. There's more to inheritance than your genes. Not all inherited traits are dependent on DNA sequence. Inheritance that is not encoded in DNA is referred to epigenetic. Okay, so this is where I'll leave you. These are the boys now. We're still watching them. And I feel always bad because I also have a daughter. And so, I know poor Siri, 15-year-old, and I know what she's dealt with through her life. No, I have a wonderful daughter, Siri, 15, and the two boys right now are 10. And we always tease Siri, had the boys been born first, she may have never existed because, it's a lot of work. All right, so any questions?
Thank you so much for this. I have a microphone that I'll pass around so that we can all hear everybody's questions. But I'm going to go first. No, I'm not. I'm going to the boss. Hello, Laura.
Hi, Mark. Thanks. So when you were originally talking about some of the twin studies and stuff, I was thinking about some of the really controversial twin studies that they did in I want to say 60s, 70s?
Yeah.
Is there anything, what do you think about that? And is there anything good that has come of that? Because it's all to me, it's all really heartbreaking, the things that they had put, twin children through. And is there anything that you've seen that's been beneficial from that?
Honestly, Christine would be the person to ask that question of, I would think, more than myself. I mean, what I'm thinking of is, there was a Netflix documentary with the triplets. Did you see this? The triplet boys. And supposedly they purposely separated them when they were born and put them in 3 completely different families. They didn't know about one another, but they ended up, 2 of them met each other in college, and then they found the 3rd one. And it was...
Did one of them end up taking his life?
I believe so.
Yes.
Yeah. The ethics is just horrendous.
Scientist. What are you going to do?
Absolutely, yeah. No, and right. I mean that's something that, again, I'm not aware of the details in history, but there are definitely documented cases where they did those horrible separations. But then when they, as far as just that documentary that I saw, when they did reunite, they were just amazed, right, at some of the things, the traits that they had in common. Yeah. Yep. That's all I've got. Yeah. Mm-hmm. Yes. Oh.
Hey, great
Hi.
presentation.
Thank you.
I love the epigenetic stuff. It's fascinating. But I want to make sure I understand. With the inherited epigenetics, wouldn't that affect the twins the same? Because it's coming down to them the same equally,
Yes.
right?
Yeah. So if how do I do this? So you're right. So if this is like a perfect world and a perfect pathway. Yeah, the end result of either the intergenerational or transgenerational inheritance is, yes, whether they inherited it or not. In the case of twins, yeah, identical twins, they would both get it. Exactly. But fraternal, it would be very dependent. But yeah, this doesn't really address the twin issue necessarily at all. But yeah, no, you're absolutely right.
I was just wondering how exactly the epigenetics are inherited. Like, are those proteins just carried through like meiosis, or?
So yes, so it would have to be via the gamete. So either the inheritance would have to come from modifications that were made to the egg or to the sperm and the DNA that those are carrying. A lot of the epigenetic marks, when they were first studying this, they thought were completely wiped, like completely cleaned, basically, with fertilization. But now they're realizing, no, that there are whole areas that remain stable for the most part, again, from one generation to the next. But yeah, it's really about the gamete effect.
Okay. Oh, no, let's get her first, and then.
I was wondering, like, when the twins are, like, when it's prenatal, their environments would actually be different because hormones and nutrients are swimming around in the fluid, and so they're receiving things differently, and they're also probably producing their own hormones and other things, stressors that happen. So that could affect why there are certain things that are different, right?
Oh yes, definitely. Even in the case of like one placenta and one amnion being shared by twins, even then there can be, the nourishment can be differently distributed amongst each twin.
Yeah, because I know, like, not all countries recognize that the prenatal development is, like, really important, but I think in this country it's becoming, not just here, but, like, Asia too, they are focusing more on, like, the actual prenatal development is so important.
Yes.
For turning on and off markers.
Right.
And what else? I had one more thing. Can the traits that are inherited be turned off after, or are they always active unless, I don't know what would happen to, like, or traits that are turned on or turned off, can they be changed later on? Is that something?
So what I immediately think of, like, the scenario to address that question is with the smokers, and again, these known regions of methylation that occur in the smokers, where those are affecting gene expressions, they have made then a direct connection to cancer incidence.
Yeah, because I was thinking also, like, you know, with a lot of people wanting, like, weight loss or even weight gain, I guess, those things can be passed on to your children, you know, with metabolism and things
The
like
agouti,
that.
the chunky mouse.
Exactly.
Yeah.
So, I mean,
right.
I guess a lot of people are looking into that, too, because
Yeah.
there's a big market for it. But
It's hard because, right, some traits are relatively straightforward. Other traits are such crazy combinations, right, of events of other gene expression. It's not an easy path to trace, you know.
Yeah.
But certainly, I mean, you know, as far as I think that cancer linkage and that not being eliminated, but again, them being able to map that back to that specific genetic region, I think that says that there's potential, right, to change a trait.
Just wondering, like, because it seems harder to do it in a natural way as opposed to, like, with the mice, they're doing it synthetically.
Right. Right. Half the battle is finding a good gene to study. Yeah.
So I'm just wondering if, so if you plan to have kids, like, if a mother is planning to have kids maybe in like a couple years, would nutrition and like all of these factors be only effective after conception? Like,
No.
you would want to like watch what you're taking in
Right.
when you're pregnant, or is it like years prior, like
Yeah.
your whole lifestyle?
they recommend if you're of reproductive years and you're sexually active, that you should be, taking a folic acid, a support basically diet. And so, yeah, it's got to be preemptive. It's got to be before the fertilization happens to ensure basically that you're not putting anything at in harm's way. But yeah, no, it has to be beforehand. Yeah. And folic acid is one of the positive epigenetic diet staples.
So before we do our final wrap up, I want to ask everybody to consider helping the library do assessments on our outreach sessions. So as you walk out, you'll see two of these QR codes, three QR codes at each exit here. If you're willing to help us out, please respond to our survey, or you could do it this way. Way to lead the group. Natural leader there. So thank you for considering doing that. I have two final questions. One, you were talking about how when the mothers take care of their kids, then those kids end up activating good genes and they have happy children and stuff. And then when they neglect them, they have sad kids. And
Anxious.
I felt a little. Well, no, I want to understand. Excuse me, I believe I have the mic right now. So, so were you implying that only the mother has influence on those kids, or does the father have some?
Oh, yeah, no, I did not mean
Right.
to go there.
So, like,
No,
if
I
they're
think
a
it's
loving
if
father and
they're
they care for their
nurtured.
kids,
Yes,
they can also.
if they are nurtured. Well, didn't they do and again, poor Christine, I keep looking, didn't they, you've done studies with rats, right? And not having, in that case, it would be like not a nurturing thing, but just social interactions, right? And I know that they've definitely looked at that before, where they would not allow again, these are lab animals, to be in the cage with the mother. And then they've looked at the deficits, right? That's clearly a thing. But yeah, no, I
But
don't
no one
believe
studied the father
it
part?
matters where it's coming from. Yes,
Yeah,
just as
just-
long as they're
They just
being
don't care.
nurtured.
Yeah. Okay. So final question, and I believe this is a very positive thing that I got out of here. Now, I'm not a smoker because of all these negative things and all these bad things, but what I believe I got out of your talk today is that we could all start smoking today, and as long as we, quit in a couple of years,
Right, yes.
all of that damage to our genetics is going to go right
Yeah.
back to normal. That's what we learned, right?
Right.
So,
No, probably not a good idea. Yes. Not,
I'm
not
thinking,
a good
whoa, let's,
Uh-huh.
I could lose some weight. I could be cool.
Yeah, no, I would not advise. There's a lot of other layers that
Oh,
are
okay.
going on. Yep, absolutely.
Okay. All right. Well, hey, with that, I want to thank everybody for coming out. Let's give Dr. Britt Ranson Olson a hand.
thank
Hopefully,
you.
she'll be willing to come back and do another talk sometime in the future. She is kind of hard to pin down here. So I feel. No, maybe
Yes,
even bring your
cool, yes.
daughter, and we could talk about how awesome
right,
daughters
right,
are.
Absolutely.
So, okay.
Oh,
Thank you very much.
Very much. Yeah, thank you. Appreciate it.